All right, so now I want to talk about the T cell receptor. So T cells, of course, are part of our acquired immune response, and T cells come in two types, CD4 and CD8. Their T cell receptors are going to be very similar in how they do somatic recombinations. And if you've already watched the lecture on B cells, you'll understand what somatic recombination is, but I'll go over it again here because we're talking about T cells. So when we talk about diversity within the T cell receptor, we are talking about in the ability to bind to peptide antigens. So the T cell receptor binds peptides, and these peptides are associated with MHC molecules, the major histocompatibility molecules, which we'll talk about in a different lecture. So the diversity we want to generate is the ability of your T cells to bind to any potential pathogens, peptides. Peptides, of course, come from proteins, and pathogens are made up of many proteins. And so we have to generate a diverse repertoire of our T cell receptor. Like our B cell receptor, our T cell receptor diversity begins in the germline. And this is where you get the genes from your maternal and paternal passed on to you. In the germline, you have both alpha and beta T cell receptor gene segments. And a T cell receptor, in its very basic look, it's always going to be associated with a membrane. And so you're going to have, so you can think of it, I'm just drawing it as boxes. You have alpha variable, beta variable, alpha constant, and beta constant. And so that's just basically the general structure of a T cell receptor. So your alpha and beta gene segments are going to form the variable region that will ultimately bind to a peptide. So here's our peptide. And it's that variable region that is going to hold the diversity, whereas the constant regions, of course, are going to be constant from T cell receptor to T cell receptor. So the diversity is with gene segments at the level of somatic recombination. And somatic is simply anything not germline. Germline means it's coming from eggs and sperm. Somatic are your other cells in your body, and these are your T cells, so your T cells are somatic cells. And so somatic recombination of VDJ regions within the alpha and beta gene segments, and V stands for variable. D is for diversity, and J is for joining. And so these VDJ regions are going to recombine to form various binding sites for different peptides. And they are going to have the ability to recognize potentially all known peptides for pathogens that you may be exposed to during your lifespan. So these recombination events will be followed by alpha and beta chain polypeptides. And then, of course, these will assemble into your T cell receptor, or your T cell receptor, TCR. Okay, so before we continue on this, I want to just discuss a little bit about the differences between the T cell receptor and your antibody structure. There are similarities, but they are very different in many key ways. So when you talk about a T cell receptor, we'll put the T cell receptor over here, and we'll talk about a B cell receptor over here. A B cell receptor can become an antibody. And so this is, of course, secreted. And this is, of course, membrane bound. A T cell receptor is always membrane bound. So you do not secrete T cell receptors. Now, T cell receptor kind of is given a look um, kind of like this. And your B cell receptor is more of a Y-shaped with two arms. And two binding sites. I'm going to move this little guy down. Okay, so now they look a little bit similar, but I'm going to show you right now how they're different. This is your peptide binding site. And it only has one. And over here, this is your antigen binding site. And there are two. Okay, so T cell receptor always membrane bound. T cell receptor only has one peptide binding site, whereas the B cell receptor has two antigen binding sites and can be either receptor bound as a B cell receptor, or it can be secreted as an antibody. Now your T cell receptor has two halves, and your antibody has two chains. So here is our light chain, and here is our light chain, and then these are our heavy chains. And our heavy chains go all the way through the trunk. So we have this region here, which is known as the FC region, and this region here, which encompasses both the heavy and light chain, is known as the FAB region, or the fragment where antibody binds, and the FC is the fragment crystallization. So it was a process of isolating the antibodies, and they got that name. Now on your T cell receptor, you have alpha chains and beta chains. So here's your alpha variable, so put a V, and we have our beta variable, and we have our alpha constant, and we have our beta constant. And so our constant regions, of course, will be what's embedded in the T cell. So here's our membrane. So the T cell receptor structure is actually complementary to the Fab fragment of your antibody. So this Fab fragment is complementary to the T cell receptor, because keep in mind there's one binding site on each Fab fragment. There's one binding site on the T cell receptor. The Fab fragment has a variable light region, constant light region, so I can add that. So this is going to be light variable, light constant, heavy variable, heavy constant. And so that kind of similar to the alpha and beta variable and constant regions. So they're similar in that way. But again, they have differences too, which we've already discussed. Okay, so now let's talk about the genes of your T cell receptor. And we'll talk about the different T cell receptor molecules. All right, so with respect to your T cells, you have your alpha and beta chain loci. So we have our beta chain loci, and you have our alpha chain loci. And so similar to our antibodies, your beta chain has diversity genes and your alpha chain does not. This is similar to the heavy chain on your antibody having diversity and your light chain do not have diversity. You have, again, a leader sequence. and this leader sequence is what helps to get the messenger RNA properly processed into a protein. So it takes the messenger RNA from the nucleus and gets it into the endoplasmic reticulum, etc. You have variable regions. So this is going to be variable beta 1. And then you can have up to 50 beta variable regions. So variable beta 50. And then you're going to have diversity genes. And so you have diversity beta 1. And the diversity genes are different in your beta loci than they were in your antibody. They're not connected, and there's only 2. So you have a diversity beta 2, but it's down further in the gene. Now next to diversity beta 1, you'll have your joining genes. So you have joining beta 1, and then you can have up to 6. And then after beta 2, I'm going to have to move beta 2. It's too close. We have 6 of these guys. And then we have our constant. And this is constant beta 1. Then you have your diversity beta 2. And diversity beta 2 is again going to be joined with this longer. We have our joining 2s. And again, there's going to be 6 of those. And this is going to be followed by another constant region. And it's the constant 2, beta 2. Okay, so that is how the beta chain loci is. And then the alpha chain, similar to our light chain, do not have the diversity. So our alpha chain loci, and I should mention beta chain is going to be on chromosome 7. And the alpha chain is going to be on chromosome 14. And so our alpha chain will have, of course, our variable regions and our leader sequences. And I should have put a leader here, sorry. Squeeze them in there. We'll have our leader, we'll have our variable alpha, and then the alpha can have anywhere from 45 to 80. So I will put our leader sequence, and then I will put variable alpha 80. And then we're going to have our joining genes, and there's up to 50 joining genes. So this is joining alpha 1, joining alpha 50. And then they have a constant alpha region. Okay, so that is the alpha and beta chain loci. And so similar to our B cell receptor, we're going to get rearrangement of these alpha and beta chains to form the T cell receptor. So the T cell receptor rearrangement, and in this case we're in the thymus to do the T cell receptor rearrangement. And again, we're going to have our different loci. So we'll take this beta chain, and we'll bring it over. And the beta chain, we are going to basically recombine the DNA. and we're going to get a variable region. Um, we're going to get your joining region with your diversity region paired up, and then that will pair up to a variable region. So one is DJ rearrangement. And so you'll get your D beta 1, and we'll say joining beta 6. And then after that, you will get your V recombination. And so you will get your leader, you'll get your variable beta 20, and then they're going to bind to your diversity and your joining gene. And here you have your constant regions. So you can have your constant beta 1. And then this will become your messenger RNA, and this will become your beta portion of your T cell receptor. So you get your messenger RNA, and then you have splicing events, and then you have everything come together. Etc. Same that happened in our B cell, nothing different. Okay, so now you have your message and you make your beta. Chain protein. And then you're going to have the same rearrangement with your alpha cells. So your alpha, so this is for your beta chain. And then I'll just write very simply for your alpha cells. Alpha cells, you're going to get recombination of your variable and joining genes, and then they will combine with your constant alpha. You get constant alpha, and then you get your messenger RNA, and then you get your alpha protein. And then together, both of these will make your T cell receptor. Okay, so with respect to T cell receptor, let's talk a little bit about diversity. So diversity is going to be multiple variable genes. So this is germline diversity. And then you're going to have junctional diversity, just like with your B cells. And so this is when you're joining your VDJ regions, you get the addition of nucleotides. And this is done by an enzyme called the terminal deoxynucleotide transferase. Also known as TDT. And this deoxynucleotide transferase, adding random nucleotides in between the diversity, joining, and variable regions, is higher in T cells than in B cells. It's about 10 times higher. And then you also get random combination of alpha and beta chains. So every alpha chain made can combine with any beta chain made and vice versa. An important difference between antibodies is that the T cell receptor never changes once it's made. B cell receptors, or antibodies, can undergo somatic hypermutations. Let me rewrite that underneath. So it's called somatic hypermutations, which we'll talk about in a different lecture. Okay, so with respect to T cell receptors of antigen recognition, it's pretty straightforward. I put this little figure here because I cannot draw a three-dimensional structure, but this is really how the T cell receptor looks. You have in here, in the middle here, a peptide, and it binds to an MHC molecule, and your T cell receptor on this side is going to interact with not only the peptide but also with the MHC molecule. So if you draw this out, you always will have a T cell, and on the surface of the T cell will always be this T cell receptor, which has the alpha and beta chains, and the T cell receptor will always have a sort of a peptide binding groove, whatever that might look like. It'll be different for every T cell receptor. And on the other side, you have to always have an antigen presenting cell, an APC, or some cell expressing an MHC molecule. And so you have your MHC, which looks like a little cup. And the MHC is membrane bound, the T cell receptor is membrane bound, and the MHC molecule is going to have a peptide that the T cell receptor will recognize. What's important though is that the T cell receptor is also going to recognize the MHC molecule. And I didn't draw it well enough here, so let me make that a little more clear. So the T cell receptor will actually touch your MHC. So TCR recognizes MHC. Okay, and so only if the T cell receptor recognizes the MHC and only if the T cell receptor recognizes the peptide can it be activated. So it takes two recognition steps. One is MHC and two is peptide. Okay, that doesn't mean the T cell is going to get activated, but in order for a T cell to even think about getting activated, these two steps have to come into play. Okay, so there is one special case though. It's called toxic shock syndrome, and I'm bringing this up here because I just drew this out that you have to have the MHC recognition and a peptide recognition in order for the T cell to get activated. So in the case of toxic shock syndrome, why it's very different is a toxic shock syndrome is caused by bacteria superantigens. And this is by our common bacteria of Streps or Staph. So what these guys can do is, let's say, for example, here's our T cell receptor, and we have a T cell receptor that recognizes a peptide that looks like this. We have another T cell receptor, and this T cell receptor recognizes a flat peptide. We have another T cell receptor, and this T cell receptor recognizes a square peptide. Okay, so we have three different T cells, three different types of T cell receptors, and we have an MHC. I'll draw an MHC. Here's MHC. Oh, sorry, this is our antigen presenting cell, APC. And our antigen presenting cell will have the MHC on its surface. It will be interacting with all of these different T cell receptors that have different specificities for peptides. And a bacteria superantigen will come along and it will glue these together. It doesn't even have to bind to the peptide binding site. So here's our superantigen, superantigen, superantigen. And we're going to say our peptide is going to be a diamond. It doesn't fit any of these T cells, but because the superantigen is present, all the T cells will get activated. And it's the T cells sort of tricking, the superantigen is tricking the T cells to think that they've seen their antigen. So you get mass activation of T cells. And this is the CD4 type. And you get cytokine storm, which leads to shock, which can lead to death. And so this is a special case because nothing else can really do this. They can't get a wide activation of T cells non-specifically. It doesn't happen except for when you have an antigen that can kind of trick your immune cells into thinking that they are seeing their particular antigen or their particular pathogen. Okay, so what really has to happen for a T cell to get activated? Well, the first thing we have to talk about is not only the T cell receptor and not only the MHC molecule, because T cells have a little bit more stringency about getting activated. And the reason is because once you activate a T cell, you can't unactivate it. So you really want to make sure that you want this cell activated, or else you're going to get things like autoimmunity. Okay, so we have a cluster of molecules on the surface of T cells. So T cell surface molecules cluster. Oops, what happened there? And they're given certain names. So again, we'll have our T cell, and we have our membrane. So here's our T cell, and there's a cell membrane. Now we already know that we have a T cell receptor. And this T cell receptor has a membrane bound as well as an external part. This is called the TCR. Now associated with the TCR are going to be several molecules. So some of the important molecules, I'm just going to draw it as a box, is CD3. And CD3 is cluster differentiation number 3. And CD3 is what actually signals if your T cell receptor gets activated. It has tyrosine kinase motifs that will signal, which we'll talk about later. Your T cell can also have a CD4 molecule. CD4 molecule looks like almost an F. It has 1, 2, 3, 4 parts, and so this is called cluster differentiation 4 molecule. Or your T cell can have cluster differentiation 8 molecule, which looks just a little bit different. 8, oops. We're going to have 2 parts to this 8. Get rid of that part. Okay, so this is cluster differentiation 8. And then you're going to have a series of what we refer to as adhesion molecules. But the T cell receptor alone cannot activate a T cell. The T cell receptor has to be associated with either a CD4 or CD8 molecule, a CD3 molecule, as well as adhesion molecules. And really the adhesion molecules are there to make sure that the antigen presenting cell and the T cell stay in contact. If the adhesion molecules are not able to do that, the T cell and the antigen presenting cell cannot stay in contact long enough for the T cell to get activated. And so these molecules are things like CD11A, which is also known as LFA1. All right, so now when a T cell gets activated or it sees its antigen and MHC complex, it forms something called the immunologic synapse. So the immunologic synapse is made up of 2 cells. The T cell plus the presenting cell. Okay, so we have over on this side, we'll have our macrophage, kind of blobby. And then over on this side, we'll have our T cell. So here's our T cell, and here's our macrophage. And I'm actually going to draw this guy a little closer. So here's our blobby guy, macrophage. Okay, and we have our T cell. Now we're going to say this is a CD4 T cell. And a CD4 T cell, of course, has CD4 on its surface. So here we'll have our T cell receptor. So here's our TCR. And associated with our TCR will be our CD4 molecule. On the opposite side, we have our MHC molecule. Okay, and in the middle of the MHC molecule is some peptide that the T cell reacts to. Okay, so the T cell and the MHC molecule are interacting with each other. So what has to happen is we have to have our CD4 cell has a transmembrane domain, and then it has our 4 and it's the 4th one that binds to the MHC. So CD4 binds MHC. And it binds an MHC called MHC class 2. And so you can see that it will bind here. And now once you have MHC class 2 binding to CD4, you have the T cell receptor binding to the peptide. You now have a tight adhesion, but it's not quite tight enough. You also need to have a certain other adhesion molecules. So on the T cell, you will express an adhesion molecule called LFA1, and it'll bind to an adhesion molecule on the antigen presenting cell called ICAM1. So those 2 will interact. Now you also have to make sure that you want this cell to become activated. So the antigen presenting cell has another molecule it expresses, and it's called CD80. And your T cell on the other side has a receptor for CD80 called CD28. Okay, so what is going on with CD80? CD80 is important. CD80 means that this macrophage has seen a foreign antigen. Okay, so the macrophage ate the pink bacteria. Its toll-like receptor on the surface, TLR5, got activated, for example, because maybe the bacteria had a flagella. So here's TLR5, and here's our little bacteria that has a flagella. And this activates our macrophage, and activation of our macrophage causes CD80 to be expressed. And CD80 is telling the T cell that this macrophage is activated. You're seeing this peptide. I'm telling you by CD80 that you need to turn yourself on and start working. And so in order to turn itself on and start working, of course, excuse me, the TCR has to be associated with CD3, and then CD3 will send the signal to activate your T cell. Okay, so again, first thing that has to happen is your T cell receptor has to interact with MHC, has to check if the peptide matches. CD4 then glues that together by binding to the T cell receptor and MHC, and then you get the adhesion molecules gluing it together even tighter, saying these guys need to hang out and make sure they have to be activated. Your APC, if it recognized that the antigen actually came from a foreign antigen, will be activated and express CD80. CD80 will tell the CD4 T cell by binding to CD28 that this is something foreign and you need to get activated. So what these are called are signal 1 and signal 2. So here's signal 1 and here is signal 2. So signal 1, T cell recognizes peptide, plus MHC, and then signal 2 is that APC is activated by foreign antigen. So both signal 1 and signal 2 must be seen in order for the T cell to be activated. Okay, now there is one other molecule that's going to be important. There are a lot of things that are going to come up, and I'll draw one more in here for another story. So once the signal is given to activate, your T cell, oops, how does it do that? Your T cell will express CD40 ligand, which is CD154 or CD40 ligand. Okay, so we'll draw a CD40 ligand as a little box. And your macrophage will have CD40. And they will interact. And this will send a message to your macrophage, and it's called the help message. And the help is going to help the macrophage kill the bacteria. So macrophages are decent at killing bacteria unless the bacteria, I'm using bacteria as an example, has the ability to evade the immune mechanisms. And so by activating the CD4 T cell, the helper cell, remember this is a helper cell, you're going to signal the T cell that this macrophage is infected with something, it needs help. The T cell will upregulate CD40 ligand. The CD40 ligand will signal to the antigen presenting cell, hey, you're infected, you need to kill whatever's inside of you. And the macrophage will kick in even more sort of anti-pathogen molecules in order to try to get rid of whatever it is infected with, whatever this bacteria with flagella is. And so that's kind of what's happening. So the T cell gets activated by signal 1, signal 2, and then the CD4 T cell is able to give that help signal back to the macrophage to help it become a better killer of this bacteria. Okay, so just a little bit more about your T cells that are different from your antibodies. So and this is just very vague, we'll go over this in more detail in future lectures. So we have T cells here, and we have our B cells, which we just talked about in the prior lecture. So our T cells are going to come from the bone marrow, the lymphoid progenitor cell, which of course came from the pluripotent stem cell. And the B cells also in the bone marrow, lymphoid progenitor cell. And so if you're going to become a pre-T cell, you're going to migrate to the thymus. And this is where you do the VDJ rearrangement. B cells will stay in the bone marrow, and this is where they do their VDJ rearrangement. Okay, so they both do VDJ rearrangement, but they stay in different cell locations. Your T cells will migrate to the thymus, that's where they get their name T cells. B cells will stay in the bone marrow. It's not where they get their name. They get their name from the bursa fibrosa from birds, where B cells were first identified. It just so happens coincidentally that in the bird, B cells matured in the bursa fibrosa, where they got their name, and it just happens that they develop in our bone marrow, so the B works. Okay, so that's all I have about T cells and T cell receptor for now.